Morning everyone. Okay, so in GIT, after stomach, the next part which we have to discuss, that is the small intestine and large intestine. Okay, small intestine and large intestine. Now, small intestine, large intestine, both they are the part of GIT. Okay, that means those four basic layers will be present. Okay, those four basic layers will be there. Now, what are the four basic layers of GIT? Yes, mucosa, submucosa, muscularis externa, and serosa or adventitia. Now, what are the layers forming the mucosa? First, lining epithelium, lamina propria, muscularis mucosae. Okay, these are the three layers. Okay, so all those layers will be present over here, but there will be some modification. Okay, because each and every part separate role is there, that's why modifications are present. Okay. Now, in case of small intestine, we know that the main function is it will complete the digestion and absorption start from small intestine. Okay. Now, for absorption, some modifications are given in the small intestine. First modification is extensive length. If you see the length of the small intestine, it is approximately 6 meter. Okay. So, that extensive length helps in absorption. Okay. Next, another modification is there, which is in the form of circular folds. Okay. If you see the interior of the small intestine, at that time you notice that circular folds are there. Okay. And what are the name of those circular folds? Plica circularis. The circular folds are called as plica circularis, or also called as valves of Kerkrings. Okay. Now, why these circular folds are present? Not exactly. Actually, what they will do, they will reduce the speed. Okay, whenever anything is traveling through it, it will reduce the speed, so that more time is there for absorption and digestion, complete the digestion. Okay, just imagine you are driving in a road where so many speed breakers are there, so you can't drive fast. You have to slow down your speed. Okay, these things are also same in case of small intestine. Now, in case of small intestine, the plica circularis which are there, these are permanent circular folds. Okay, these are permanent circular folds. The plica circularis or valves of Kerkrings which are there, these are permanent circular folds. What is the meaning of this permanent circular fold? At any time of point, these folds are present. Okay, doesn't matter whether food is there or not. Okay, the circular folds will be there. Now, why it is permanent? Because you can see all the layers of mucosa, that is lining epithelium, lamina propria and muscularis mucosae, they are extending into the circular fold, and in some cases submucosa also extend into the fold. Because of this reason, the folds are permanent in nature. Why I am mentioning? Because in case of stomach, we have seen that the gastric rugae which are present, these are not permanent, these are temporary. As the stomach distend, those rugae disappears, but here it will not disappear, it is permanent. Okay, because of this reason, as I have mentioned, this is permanent. Next, if you see the plica circularis, okay, the number of plica circularis, as you go from duodenum towards the ileum, then the number of plica circularis reduces, and the space between the plica circularis, it also reduces. And in case of terminal part of the ileum, this is absent. Okay. Next, another important modification which is present inside the lumen, okay, or you can say inside the small intestine, that is the presence of villi. Okay. We have discussed the structure of villi. I have shown you the structure of villi, what is there inside the villus, how the absorption is taking place, all those things we have discussed. Okay. So, the villi is present, which is finger like projection. What is the main function of the villus? Mainly it will help in absorption. Okay, because जो villi होता है, direct absorption through villi is taking place. For increasing the surface area, we are having extensive length. Okay, after that we are having microvilli, those will increase the surface area. जो villi होता है, directly it will absorb. Okay. Now, if you remember what we have discussed about the villus, okay, I don't know whether you have studied or not. If you see the villi, they are present in case of duodenum also, they are present in jejunum, and they are present in ileum. Okay. Structure and function remain same, but what is the difference? The difference is in the shape, difference is in the number, okay, difference is in the size, shape and number. These three things will differ, but other things, structure, function remain same. Okay. So as you go from duodenum towards the ileum, what are the changes taking place in the villi? The first part of the duodenum, they appear as broad ridges. Okay. Next, they become tall and foliate like in the distal duodenum and proximal jejunum. Further, if you go, they gradually shorten to form finger like in the distal part of the jejunum and ileum. So these are the changes taking place, that also in the shape and the size. That means height will reduce, the shape will change, but the structure function remains same. Okay. So why this is important? Anyone? Why this is important? Why I am mentioning? Because villi to villi a function same hi rahega, absorption. Then why I am mentioning? Any idea?
Because it is present over the folds of.
It is present, I know, but what is the significance? Think histologically. What will be the significance? No. Because whenever we will see the slide under microscope, okay, whenever we will see the slide under microscope, तो small intestine का कितना part होता है? How many parts are there in small intestine? 3 parts are there. Okay, so if you see the villi of the duodenum, if you see the villi of the jejunum and ileum, okay, सबका size and shape same नहीं होगा, it will be different. From there we can identify. Okay, that's why I am mentioning that as you go from duodenum towards the ileum, the size and shape changes. Okay, all are having not equal size and equal shape. Next, another important point is, as you can see, the density of the villi, it also changes, ranging from 10 to 40 per square millimeter. Okay, this is the range. Okay, or you can say the number of villi per square millimeter. And if you see the length of the villi, the height, it is 0.5 to 1 millimeter. That means these are not microscopic. Villi can be seen macroscopically. Okay, villi can be seen macroscopically with the help of magnifying lens. That means you have to open the small intestine, especially the duodenum. Okay, have you seen the interior of duodenum in the dissection hall? Yes, those plica circularis, then major duodenal papilla, minor duodenal papilla. Yes. So at that time you could have done one thing. You could have taken a magnifying lens and you could have focused on that particular part. ठीक है, तो finger like projection दिखाई देता है छोटा छोटा. Okay, it can be seen. I don't know about the specimen which is available over here, but in a fresh specimen it can be seen. Okay. So so this is villi, one type of modification. Next, another modification is the lining epithelium which is present in the small intestine. Okay, on the surface of the lining epithelium a modification is there, that is the microvilli, that also striated border, and because of this microvilli, okay, it further increases the surface area for absorption. Okay, so this is also a modification. Do you remember the lining epithelium of stomach? Stomach we have discussed in the last week. Do you remember the lining epithelium of stomach? क्या है? What is the lining epithelium of GIT? Only 2 areas are there where exceptions are present. Simple columnar epithelium. Okay, simple columnar epithelium, that is the lining epithelium of GIT. Several times I have mentioned in the class. Except the esophagus and lower end or terminal part of the anal canal. Okay, the simple columnar epithelium is not there. Remaining everywhere you are getting simple columnar epithelium, as simple as that. But there will be some modification. Okay, so in case of stomach we have seen only simple columnar epithelium, that also mucus secreting cells are present. But here, okay, along with the simple columnar epithelium we are getting microvilli. So that is a modification in case of small intestine which is absent in stomach. Okay, so these are the small points which you have to know or else it will be very difficult to differentiate the slide, because during the time of exam you can't say that I have studied this, I have not studied that. Okay, ऐसा नहीं चलेगा. Whatever slides are put on the microscope, you have to identify. Okay, next, last week I hope you remember we have discussed about the gastric glands. How many types of gastric glands are there? 3 types of gastric glands: main gastric gland, cardiac gland and pyloric glands. ठीक है, तो at that time what I have mentioned, that pits are there, the depression, the pits, and deep to the pits what are present? Gastric glands are present. ठीक है, तो same in case of small intestine also, depressions are there, and here these are called as crypts, not pits. Crypt, C-R-Y-T-S, crypt. Okay, C-R-Y-P-T-S, crypts, and deep to the crypts we are getting intestinal glands, and these are also called as crypts of Lieberkühn. Okay, so in case of stomach also we have seen glands are there. Here also glands are present, especially in case of lamina propria, but the function of the glands are not same, it is different. Cells which are present in the cell glands are different. Okay, so see, you have to correlate along with the stomach. Now in this diagram, okay, there are 2 diagrams. Okay, now in this diagram you can see this is the interior of the small intestine, where this is the lumen of the small intestine. Okay, now from this lumen you can see the finger like structures are projecting. These are called as villi. Okay, these are called as villi. Now in between the villi, can you see small spaces are there? Can you see small spaces are there? These are called as intervillous space. Intervillous space. Okay. Now suppose this is one villus, this is one villus. If it is single, then it is called as villus. Multiple, then it is called as villi. Please keep that in mind. Okay. Now if you take one intervillus space, so this is one villus, this is another villus, in between that the area which is there, this is called intervillus space. Okay. In intervillus space you can see the opening, so this is the opening of the crypt, opening of the intestinal crypts. Now if you go deep to the crypts, then you can see the gland, intestinal gland, are also called as crypts of Lieberkühn. Okay, where different types of cells are there and secretions are taking place. Okay, so this particular part will be the lining epithelium. Okay, now this is the lamina propria, as you can see. If you go below lamina propria, I hope you can see the muscularis mucosae. ये 3D diagram है, ठीक है. So this is the muscularis mucosae. If you go below muscularis mucosae, here you can see the submucosa. Okay, this is the submucosa, and below that muscularis externa, and then you can see the serosa also. Okay, so if you compare with the stomach, what do you think? What is the main difference? In case of stomach, you will not see any villi over here, right? In case of stomach, villi are absent, but here you can see villi are present. Okay, now another important point which you have to know, that is the lining epithelium. Okay, जो lining epithelium है, how they are oriented, how they are placed, because along with the lining epithelium we are also getting the microvilli having striated border. Okay, yeah, I hope you remember this diagram. That day I have shown you this diagram last Saturday. Okay, now here you can see the lumen of the small intestine. In the lumen of the small intestine you can see this is one villus, this is one villus. Okay, in between both the villus you can see the area which is there, that is called as intervillus space. Okay, now if you go below lower part of the intervillus space, you can see the depression, this is called as crypt. Okay, the opening which is there, that is called as crypt. Deep to the crypt, what will be present over here? Glands, intestinal gland, are also called as crypts of Lieberkühn. Okay, now just see how the lining epithelium is oriented. Now this lining epithelium is covering the external surface or external part of the villi, external part of the villus. That means if you take one particular villus, ठीक है, villus का जो external part होगा, that is covered by the lining epithelium, which is simple columnar epithelium, and top of that we are getting the microvilli. Okay, so if you zoom this particular part, see this columnar epithelium is there, and over that we are getting the microvilli, that also striated border. Okay, and in case of small intestine, the cells, especially the lining epithelium, they are called as enterocytes. Okay, they are called as enterocyte. Okay, so don't get confused. Enterocyte का मतलब क्या है? Lining epithelium. Next, so here also you can see this is the villus, this is the inner part of the villus, this is the outer part of the villus. In the outer part we are getting the lining epithelium, and over the lining epithelium we will be getting microvilli, that also striated border, which helps in increasing the surface area for absorption. So this is how the lining epithelium is oriented. Okay. Next, if you take the example of overall small intestine, okay, then as I said, those 4 basic layers are present. Now, in those 4 basic layers, what will be the first one? Lining epithelium, that means mucosa. In mucosa, 3 layers are there forming the mucosa. So, what will be the first one? Lining epithelium. Okay. Now, in lining epithelium, already I have mentioned what is present? Simple columnar epithelium, that also having striated border. Okay. Next, the lining epithelium, along with the lining epithelium, other type of cells are also present, because just now I have shown you the intestinal glands, or also called as crypts of Lieberkühn. ठीक है, तो अगर gland है, inside the gland different types of cells will be present. ठीक है, so here you can see the list of cells which are present in the various part of small intestine. Here I am mentioning about the small intestine only, not about the large intestine. Okay. Now, few of the cells which are present along with the lining epithelium, and other cells are present inside the gland, intestinal gland. Okay. Now, if you see the enterocyte, goblet cell, these are placed together along with the lining epithelium. Okay. Now, already we have discussed, if any goblet cells or if any mucus secreting cells are there, how you will identify them under microscope? Yes, it will be white in nature, white in color, because they appears empty. Okay. See, these things are important. I don't know what you guys are doing. This is not the first time. From respiratory system onwards, I am mentioning same thing. ये जो गॉबलेट सेल्स है या कोई भी म्यूकस सिक्रेटिंग सेल है, तो H&E स्टेनिंग में कैसे आइडेंटिफाई करोगे? हाउ विल आइडेंटिफाई? वाइट कलर का अपीयरेंस होगा. वाई वाइट कलर का अपीयरेंस होगा? बिकॉज इट एम्प्टी, बिकॉज जो प्रोसेसिंग होता है सेल प्रिपरेशन में, टिश्यू प्रिपरेशन में, ये वॉश हो जाता है, वॉश आउट हो जाता है. तो इफ इट इज वॉश्ड आउट कंप्लीटली, दैट पर्टिकुलर पार्ट नथिंग इज देयर टू टेक द स्टेन, दैट्स व्हाई इट अपीयर्स एम्प्टी. ओके, तो उसको स्टेन करने के लिए डिफरेंट स्टेनिंग मेथड्स आर अवेलेबल. ओके, तो फर्स्ट एंटेरोसाइट्स. व्हाट इज दिस एंटेरोसाइट्स? लाइनिंग एपिथेलियम. एंटेरोसाइट्स आर नथिंग बट द लाइनिंग एपिथेलियम. तो लाइनिंग एपिथेलियम क्या है? सिंपल कोलुमनर एपिथेलियम, दैट ऑल्सो हैविंग माइक्रोविलाई. विच टाइप ऑफ माइक्रोविलाई? स्ट्राइटेड बॉर्डर. माइक्रोविलाई में भी दो होता है, टाइप होता है, एक स्ट्राइटेड बॉर्डर एंड एक ब्रश बॉर्डर. ठीक है, दिस इज हियर वी आर गेटिंग द स्ट्राइटेड बॉर्डर. ओके, नाउ व्हाट इज द फंक्शन? एब्जॉर्प्शन एंड सिक्रेशन ऑफ सम एंजाइम्स. नेक्स्ट, गॉबलेट सेल्स होता है. गॉबलेट सेल वैक्यूलेटेड ऑन इट्स एपाइकल पोजीशन, बेजल फ्लैट न्यूक्लियस रहता है. एंड इफ यू सी द शेप ऑफ दिस पर्टिकुलर सेल, गॉबलेट सेल, वाइन ग्लास अपीयरेंस होता है, जस्ट लाइक वाइन ग्लास अपीयरेंस. ओके, गॉबलेट सेल. एंड जो एपाइकल पार्ट होता है वहाँ पे वेसिकल्स रहता है फ्रॉम विच द सिक्रेशन इज टेकिंग प्लेस. ओके, and as I said, for that special type of stain is required, that is called PAS method, periodic acid skiff method. ठीक है, it is a specialized type of staining. तो उससे अगर स्टेन करोगे then magenta color appear होगा goblet cell. Okay, next unicellular होता है and mucus secretion. The main function is it will secrete mucus. Next, these cells are very important, these three cells, that is the Penneth cell, or also called as zymosome cell. Okay, if you see that particular cell, it is completely acidophilic cytoplasm, basal nucleus, and on the apical part we are getting the zymosome granules, जहाँ से secretion होता है. Okay, now इसका main function है phagocytosis, and it secretes lysozymes and alpha defensin, and with the help of this secretion, what it will do, it will maintain the normal bacterial flora in the small intestine, which helps in digestion. Next, enteroendocrine cell. Okay, in case of stomach also we have seen enteroendocrine cells are present, if you remember. So these are specialized cell which cannot be stained with the help of HNE. For that silver salts are used. That's why they are called as argentaphene cell. Okay, that's why they are called as argentaphene cell. So these are the part where MCQs are asked. From these particular areas MCQs are asked. Now uska function kya hai? It secretes, in case of small intestine, enkephalins, vasoactive inhibitory peptides, these are secreted. So neuroendocrine cells ka function kya hai? In case of small intestine, it will control the motility, it will control the secretion, absorption, blood flow into the small intestine. Okay, all those things are controlled by these enteroendocrine cells. Next, M cells are there. Okay, these are present in the epithelial lining over the Peyer's patches. Means along with the Peyer's patches, these are present. Okay, what is Peyer's patches? So I am coming into this. Now these M cells, these are nothing but the antigen transporting cell. So they will transfer the intestinal bacterial antigen, okay, to the lymphocyte or dendritic cell. Okay. And after that, what will happen? The antibody will be produced. So if any antigen enters the small intestine, this particular cell will bind to that, and then it will be produced to the cells which can produce antibody. Okay, especially in case of intestine. So this is the function of the M cells. And lastly, the stem cells. Okay, so stem cell, already you know what are the functions of the cell. Several times we have discussed about the stem cell. So these are the different types of cells which are present throughout the small intestine. Next, you can see the diagram. Okay, so here you can see this is the enterocyte, tall columnar cell with microvilli. This is the wine glass appearance of the goblet cell. Okay, just see the secretory vesicle which are present at the apical part. Okay, this is the neuroendocrine cell. Okay, and then this is the Paneth cell. Okay, so this please keep that in mind. This particular chart you have to write in your practical notebook. Okay, along with that you have to draw this diagram also. This diagram. Don't forget. Okay, next lamina propria. So after lining epithelium, what is the next layer? Lamina propria. Okay. So if you see the lamina propria in case of small intestine, there are some areas where thick population of lymphatic aggregation are present. Okay, which are the components of immune system. For example, lymphocytes, plasma cells, these are present in some areas of the small intestine. I am not telling entire small intestine. Some areas of the small intestine, in case of lamina propria, what is present? Aggregation of lymphatic follicles or lymphatic aggregations are there. Okay, and these are the component of immune system where you can get the lymphocyte plasma cells. Okay, now these are very important, and this will constitute the Peyer's patches. Okay, this will form the Peyer's patches. Especially it can be seen in case of ileum. Okay. Now solitary aggregated lymphatic follicles, they are called as Peyer's patches, are present in the lamina propria of the small intestine, and they are forming the GALT, that is gut associated lymphoid tissue. Okay, lymphatic system till now we have not discussed. Okay, there is a chapter lymphatic system, and we will be discussing histology only. I think next week. At that time we will discuss in detail about GALT, then bulk, bronchosociated lymphatic tissue, everything. But here please keep that in mind. In case of small intestine, ठीक है, उसका जो lamina propria होता है, in some part what happens, solitary and aggregated lymphatic follicles are present. Together they are called as Peyer's patches. Okay, and these Peyer's patches are more where in the ileum. Now, and these Peyer's patches can be seen without microscope also, but with the help of the microscope it can be easily identified, and in ileum it is an important identifying feature in the lamina propria. Okay, three parts are there, so you have to understand. In case of small intestine, three different parts are there. Now from those three parts, if I keep any of the slide under microscope, and if I ask you to identify, because देखो चार layers whatever we have discussed, all those 4 layers will be present. Okay, then based on what you will differentiate. Duodenum का slide है, jejunum का slide है, ileum का slide है, based on what you will differentiate. Yeah, so for that, no, only based on villi you can't differentiate. It's not possible. only based on the villi. There are some other points also. Okay, for each and every part specific points are there, and I am coming on those points, don't worry. Now here whatever we are discussing, that is for entire small intestine. पूरा small intestine में क्या रहता है, what modifications are present, that we are discussing. After that individual one we will discuss. Okay, next muscularis mucosa, no changes. Okay, in case of muscularis mucosa, two layers are there, that is inner circular, outer longitudinal smooth muscle. Okay, now this particular muscularis mucosa help in the movement of villi and expulsion of the secretion from the intestinal gland. In case of muscularis mucosa, especially in small intestine, what is the function? It will help in the movement of the villi and expulsion of the secretion from the intestinal gland. Next, in case of submucosa, mainly loose connective tissue is present, where you can see blood vessels are there, lymphatics are there, nerves are present, and along with that we can also see collagen fibers and elastin fibers are present, okay, in the submucosa. Now due to the presence of this collagen fibers and elastin fibers, okay, it provides strength, support when the movements are taking place. For example, peristalsis movement are taking place continuously. Okay, so because of the presence of this collagen and elastin fibers, okay, it provides adequate support, elasticity and strength to the small intestine. That's why these elastin fibers and collagen fibers are present in the submucosa. Next, so this is normal general structure. Okay, but there are some modification. Now where this modification is present? The modification is present in case of duodenum. Okay, if you see the histology of duodenum, a slide of duodenum, at that time you will notice that in the submucosa it is packed with Brunner's gland, or also called as submucosal glands. Okay, जो duodenum होता है, in case of duodenum, the submucosa of the duodenum is packed with glands. And what are the name of those glands? Brunner's gland. Function क्या है? They will secrete mucus, and that mucus is rich in bicarbonate ion. That mucus is rich in bicarbonate ion. Now what is the pH of bicarbonate ion? Acidic or alkaline? Alkaline. It is alkaline in nature. If you see the pH of bicarbonate ion, it is more than 8.5 or 9 like this. Okay, so why it is required? Any idea? Maintain the, yes. So we know that the medium of the stomach is acidic, and what about the small intestine? It is alkaline in nature. Okay, so stomach से जो acidic content आता है, it has to be neutralized. For that the bicarbonate ion is required. Okay. Now if you remember about the succus entericus, which you have studied in physiology, do you remember? Succus entericus, what is the component constituent of succus entericus, what is the function, कितना secrete होता है, then you can correlate. Okay, next muscularis externa. So muscularis externa में कोई changes नहीं है, no changes are there. Okay, the muscularis externa is having two layers, that is inner circular, outer longitudinal. Okay, what is the main function? It's local mixing is taking place, and along with that it also propel the food to the next part, that is propulsion of the food from one part to the another part of the small intestine, that is done by the muscularis externa. Okay, and in case of serosa, mainly in small intestine, peritoneum is present. Only in case of duodenum you will see adventitia is present, but in most of the small intestine, what is there? Peritoneum is present. That's why serosa is there. Okay. So whatever we have discussed, that was common for entire small intestine. Okay. Now specifically for duodenum, and I will request everyone to please note this point, because after that we will go to the histo lab and we will identify the slide. Okay. So for that, these points are important. As I said, three parts are there of the small intestine. So you have to know the important identifying features separately. Then only you can identify, or else identification नहीं होगा. ठीक है. So the first part is duodenum. So what is present in the duodenum? All those four layers will be present. Okay, no changes. Next you can see villi are also there. Okay, along with that simple columnar epithelium, few goblet cells are also there, and in the simple columnar epithelium microvilli are also present. ठीक है, तो up to that much it is same, no changes. Next this microvilli are present, then crypts of Lieberkühn intestinal glands are also present, and in the glands the cells whatever we have discussed, all the cells are present. ठीक है. Next, if you see the submucosa, this is the most important identifying feature for small intestine, that is the presence of the Brunner's gland. Okay, presence of the Brunner's gland in the submucosa, that is the most important identifying feature. Okay. Now these glands are branched, highly branched tubulo-alveolar mucus secreting glands, okay, located in the submucosa. Now Brunner's glands are present deep to the muscularis mucosa. Okay. Next, muscularis externa, same inner longitudinal outer circular layer. So other things are same, okay, you will not get any changes. The only thing which you have to notice, that is the submucosa. In the submucosa it is packed with Brunner's gland. Now this is not the only point, there are some other points also. Okay, what are those other points? If you see the villi which are present in case of duodenum, first of all the length of the villi are more as compared to the other parts. That means jejunum और ileum में जो villi रहता है उसका जो length है, and duodenum में जो villi रहता है उसका length, that is more. ठीक है. Next, the number of villi are also more in case of duodenum. Okay, these are some important points. Next, if you see the lamina propria, in most of the cases pairs patches are absent. Lamina propria, what is pairs patches already we have discussed. ठीक है, जो aggregations होता है lymphoid tissue का, so that is most absent in case of lamina propria in duodenum. So these are some important identifying feature which you have to look in the slide whenever you are identifying the duodenum. Okay. Now see, in duodenum these four basic layers are present. So mucosa में क्या रहता है? Lining epithelium. Just see the lining epithelium. Along with that, goblet cells are also there. Villi are present, finger-like. Crypts of Lieberkühn in the lamina propria, muscularis mucosa. Okay. In the submucosa, see Brunner's gland, highly branched tubulo-alveolar glands are present. Okay. Now here you can't see any pairs patches over here. Okay. In this particular area, pairs patches are not there in duodenum. Muscularis externa, same two layers are there, and adventitia, in some part adventitia will be there, in some part serosa will be there. Okay. Now this is the schematic diagram of which part? Anyone? Can you identify? Who said jejunum? कैसे jejunum होगा? Duodenum. It is duodenum. क्यों duodenum क्यों होगा? Brunner's glands are there. कहाँ पे रहता है Brunner's gland? Submucosa. Okay. So like this you have to identify. So first, what is this? Villi. Okay. So this is one villus, this is one villus, and this is intervillus space. ठीक है. Now if you go below the intervillus space, ये पूरा lamina propria है. Now in the lamina propria, can you see these are the intestinal glands? See, these are the cut section of intestinal glands. Okay. And here you can't see any of the pairs patches. जो lamina propria है उसमें pairs patches नहीं रहता है in case of duodenum. ठीक है. Next you can see the muscularis mucosa. ये देखो, this is the muscularis mucosa. Now below the muscularis mucosa, what is this? Submucosa. In submucosa you can see the Brunner's gland. Okay. Now here also one particular point is there. I don't know whether you have noticed or not. जो Brunner's gland है and जो intestinal gland है, दोनों में क्या difference है? See, no. Listen. These are also glands. Okay. In classification, I know difference is there. I am not going into that. The way it appear under microscope, उसमें क्या difference है? नहीं, not size and shape. वो तो उसमें चला जाएगा classification में. Staining, yes, staining. In staining differences are there. ठीक है. तो staining में क्या difference है? What difference you can see? हाँ, तो ये mainly white appearance होता है and this mainly dark appearance होता है. क्यों? Why? हाँ, because as I said, these are mucus secreting glands. Okay, तो mainly क्या रहेगा इसके अंदर? Mucus रहेगा. So during the processing washed away. That's why it appears empty, white in nature. इसके अंदर mucus नहीं रहता है. These are not mucus secreting glands. That's why it is taking the stain and it is darkly stained. Okay, like this you have to differentiate कि कौन सा mucosa है, कौन सा submucosa है. Okay. So this is the Brunner's gland, which you can see it is packed with Brunner's gland. Next to that, what is this? This is the muscularis externa. In muscularis externa, you can see two layers are there separately, which can be seen. So this is the schematic diagram which you have to draw in your practical notebook. This one. Next, this is the histological diagram. This is under low magnification and this is under high magnification. Now I will request everyone try to identify all the layers. Okay. First, please see the villi. ये देखो, this is one villus. This is one villus. ठीक है. These are the intervillus space. If you go down deep to that, okay, you can see the glands. See, these are the intestinal glands. ये देखो, यहाँ पे intestinal glands है. Next, this is the muscularis mucosa. ये देखो, this is the muscularis mucosa. Now below muscularis mucosa, यहाँ पे क्या रहता है? Submucosa. Now in submucosa you can see these are the Brunner's gland. Okay, पूरा submucosa है where you can see the Brunner's gland. Now deep to that, what is this? Muscularis externa. Though layers easily it can be seen, and then serosa is there. Okay. Now this is under high magnification. So this is how you have to identify the duodenum. Next, after duodenum, next is the, what are the other two parts? Jejunum and ileum. Now what do you think, what will be the differences in case of jejunum and ileum? One by one. हाँ, very good. Pairs patches. Pairs patches किस में ज़्यादा होगा? इलियम में ज़्यादा होगा. कहाँ पे रहता है pairs patches? Lamina propria. Very good. नहीं, Brunner's gland absent. Brunner's gland रहता है in case of duodenum. Brunner's gland दोनों में absent रहेगा. Okay, you will not see Brunner's gland. ठीक है? See, for layer wise we have to go. So first, if you see the villi which are present in case of jejunum, okay, इसका जो length है that will be more as compared to the ileum. Number of villi will also be more as compared to the ileum. ठीक है? That is, yeah, size means I am talking about the length. Length will be more and then number will also be more in case of jejunum as compared to the ileum. ठीक है? तो ये हो गया एक part. Next, if you come below the lamina propria, pairs patches are present but the quantity is very less in case of jejunum and sometimes it cannot be seen. ठीक है? अगर रहता है एक या दो, very less. But if you see the ileum, it is filled with this one. Lamina propria is filled with pairs patches and sometimes it is extending into the submucosa. Okay? So these are some important differences by which you have to differentiate. Okay, so all the layers are present. Okay, and here in the submucosa, Brunner's glands are absent. Okay, Brunner's glands are absent. Here also Brunner's glands are absent. Okay. Next, in case of lamina propria, as I said, intestinal glands are present. Here also you can see intestinal glands, but here the pairs patches are more. Okay, pairs patches are more. Let me show you the diagram so that you can differentiate. So this is the histology of jejunum. Okay, in the histology of jejunum, first you can see this. What is this? These are the villi. So this is one villus, this is one villus, this is one villus. Okay, you can see these are the villi. This is the intervillus space. ठीक है? इसके बाद यहाँ पे क्या रहेगा? Intestinal glands. You can see these are the intestinal glands. Now here, see, lymphatic follicle are there, but it's very less. See, it is present, but it is very less. Okay. Next, this is a muscularis mucosa. In submucosa, you can't see any of the Brunner's gland. Submucosa में Brunner's gland नहीं रहता है, absent. ठीक है? और इसके बाद muscularis externa and then serosa. In case of jejunum and ileum, completely it will be serosa. Okay, so previously this is the schematic diagram which you have to draw in the practical notebook. This is the histological image, low magnification and high magnification. Okay, this is also the histology of jejunum. Okay, see the image carefully because after that you have to identify under microscope. Okay, next this is the histology of ileum. So what you have observed? हाँ, ये क्या है? What are this? Villi. Size छोटा है, as you can see. ठीक है? After that, in the lamina propria, can you see what are these? Pairs patches. So these are more in case of lamina propria of the ileum. Remaining everything is same. बाकी देखो सेम रहता है, ब्रूनर्स ग्लैंड नहीं रहता है. एंड इफ यू सी द क्वांटिटी ऑफ पेयर्स पैचेस, एज आई सेड, इट इज एक्सटेंडिंग इनटू द सबम्यूकोसा. ठीक है, ये देखो सबम्यूकोसा में आ रहा है, सम पार्ट इज कमिंग इनटू द सबम्यूकोसा. सो दिस इज हाउ यू हैव टू आइडेंटिफाई. ओके, एंड दिस कैन बी सीन अंडर माइक्रोस्कोप. Okay, so this is the schematic diagram which you have to draw, and this is for your understanding. Next, in case of please everyone please note it down in a practical notebook. After writing the duodenum, what you have to do, you have to write about the structure of villus. What we have discussed last week, and just now I have shown you the diagram. Okay, pura structure and explanation part both should be there in your practical notebook. See, I am talking about this intestinal villi. Okay, this you have to write. This is the structure and this is the diagram. Okay, after duodenum please add this one. Now I am not explaining because already we have discussed that. Okay, so after small intestine, next is the large intestine. Stop talking. In large intestine, I know you have seen the large intestine. Okay, you have studied about the external features also of the large intestine. Now, what are the three important external features of large intestine? Yes, and appendices epiploicae. Yes. So, can you see all the points over here? So, what is appendices epiploicae? Peritoneal pouches which are attached externally to the wall of the large intestine. Then what is tinea coli? Muscular, yes. Bands of which layer? Yes, I know smooth muscle fiber. Which layer? Two layers are there, circular and longitudinal. No, longitudinal layer. Okay, these are the three bands of longitudinal muscle fiber, the tinea coli. Okay, and what about the circulations or hostrations? Pouches? No. ये देखो, ये क्या है, ये जो ऐसे ऐसे करके है ना, ये देखो. These are called as circulations or hostrations. If you see the circular layer, large intestine में क्या होता है? Everyone you know that two layers are there, right? Smooth circular layer and longitudinal layer. तो इन केस ऑफ लार्ज इंटेस्टाइन जो सर्कुलर लेयर है उसका जो लेंथ है दैट इज मोर एज कंपेयर टू द longitudinal layer. So that's why the longitudinal layer is pulling the wall of the large intestine. That's why you are getting the circulations or hostrations. Okay, which is a unique important feature in case of large intestine, जो इसमें नहीं रहता है, small intestine में नहीं रहता है. ठीक है. Next, in case of large intestine, all the external features whatever you have mentioned that is present up to sigmoid colon. Beyond sigmoid colon those features are not present. I don't know whether you have noticed or not. Okay, all the important features whatever you have mentioned कहाँ तक रहता है? Up to sigmoid colon. इसके बाद नहीं रहता है. Okay, so next time whenever you go to the dissection hall, just see the specimen properly and try to identify. Okay. Next, have you seen the appendix? Appendix you have studied? Okay. So today we will be studying about the microscopic structure of appendix also. So this is the external features. Now this is about the internal structure. Now if you see the internal structure of large intestine, what do you think what you will see? Those four basic layers will be present. That is compulsory, वो रहेगा. What else you can see in the lumen? See, this is the lumen. Okay, इसमें क्या है? What is absent over here? Villi are absent. Previously we have seen villi were present, but यहाँ पे देखो villi absent है. ठीक है. Next, other things are same. What is this? See, opening of the crypts are present. Then you can see the lining epithelium. Deep to the crypts, you can see intestinal glands are present. Okay, but यहाँ जो glands है, and in case of duodenum the glands which we have studied उसमें थोड़ा difference है. Difference in the cells. यहाँ पे जो glands है mostly they are mucus secreting. Okay, so easily it can be identified. So you will see that white color portions are more in the mucosal part as compared to the duodenum. Okay. So in order to describe the large intestine, I have taken the example of colon. Now if you study the colon, all those basic four parts will be there. Mucosa, lining epithelium will be same, simple columnar epithelium having microvilli, that also striated border, and along with that goblet cells are also present. Okay, as you can see, simple columnar epithelium, that also absorptive function, along with that microvilli and goblet cells are present. Next, lamina propria filled with intestinal glands, also called as crypts of Lieberkühn. Okay, they are lined by goblet cell. Lamina propria shows discrete lymphatic follicles. Now here also lymphatic follicles will be present, okay, but discrete means the number is less. If you see the number of lymphatic follicles in the lamina propria of large intestine as compared to the ileum, it is less. Submucosa consists of dense irregular connective tissue, okay, and in the submucosa you will not see any of the gland. In large intestine also जो submucosa होता है उसमें कोई glands नहीं रहता है, no mucus secreting glands are present. Muscularis externa, same two layers are there, okay, but there is a modification. What is that modification? The longitudinal fibers form three bands and these are called as tinea coli. That you have to write or you have to mention, okay. Though it cannot be seen histologically, जो tinea coli होता है directly you have seen. Tinea coli मतलब you can't see in microscopic structure, but you have to mention that जो longitudinal fold है, that is modified to form three bands and these are called as tinea coli. Okay, and externally it is covered by serosa or adventitia. In some part serosa is there, some part adventitia, because entirely it is not intraperitoneal. Okay. Next, what is absent in case of colon? As you can see, plica circularis are absent. Plica circularis क्या है? Mucosal folds, circular folds, permanent circular folds. कहाँ पे रहता है? Where they are present? In small intestine. In small intestine. But if you see the large intestine, these are absent. Large intestine में नहीं रहता है. Okay. Next, villi, these are also absent, and then Paneth cells. As I said, the intestinal glands which are present in large intestine, only mucus secreting glands are there. Okay. All those cells are absent. So here you can see all the four layers of the large intestine. Okay, everything is almost same. Only difference is in the muscularis externa, the outer longitudinal layer is modified to form three bands of tinea coli. Okay. Now this is the schematic diagram. This is the drawable diagram which you have to draw in your practical notebook. Okay. So what you can see here. If you have noticed the histological diagram of small intestine, various part of small intestine, okay, and now you are looking at this, please try to correlate. Here you will see more number of goblet cells. Okay, ये देखो more number of goblet cells. That's why many empty spaces are there. Can you see along with the lining epithelium? So many empty spaces are there. That means more number of goblet cells are present over here as compared to the small intestine. ठीक है, एंड यहाँ पे देखो विलाई नहीं है. नाउ आई हैव सीन स्टूडेंट यूज्ड टू गेट कंफ्यूज्ड. जो लार्ज इंटेस्टाइन का लुमेन है, इट इज ओरिएंटेड इन सच ए वे दैट बिकॉज ऑफ दोज हॉस्ट्रेशंस और सर्कुलेशन, ओके, इट अपीयर्स एज विलाई. देखो विलाई जैसा दिखाई दे रहा है कि नहीं? हाँ, बट एक्चुअली इट इज नॉट विलाई. हाँ, तो आई हैव सीन स्टूडेंट यूज्ड टू गेट कंफ्यूज्ड ओवर हियर. ओके, सो फॉर दैट व्हाट यू हैव टू डू? For that you have to correlate between both the images, okay, images of the small intestine, various part of the small intestine and the colon. Then only, and several times, not one time, then only you can understand and then you can differentiate. Okay, or else if I put the slide during the time of exam, most of the student will identify it as small intestine villi. Okay, ऐसा होता है. Today only we can check. Okay, today also we can check. Most of the student will identify it as villi. Okay, so that's why I am requesting you to see properly. Or in case of large intestine, okay, layered same रहता है. In case of duodenum also, if you see the layers in case of colon, same रहता है. But as I said, in case of large intestine, goblet cells are more. That means you will see more empty spaces. Okay. Next, here in the lamina propria, as I said, the aggregation of lymphatic nodules, lymphatic follicles are very less, okay, as you can see in the diagram. Next, this is the muscularis mucosa, then this is the submucosa. In submucosa, glands are absent, mucosal glands nahi rehta, these are absent. Then this is the muscularis externa. Muscularis externa का modification क्या है? Tinea coli. The longitudinal layer is forming 3 bands, that is called as tinea coli. Okay. Next, this is the this is under low magnification, this is under high magnification. Okay. So please try to identify all the layers. Next is the histology of appendix. That means the features of the large intestine will be there. But what extra thing you can expect in the appendix? What additional part you can expect in the appendix? Those 4 basic layers, everything are same. So extra क्या दिखाई देगा? See, everything is same. Appendix, in case of appendix, mucosa, lining epithelium, simple columnar epithelium, numerous goblet cells are there, striated border, lamina propria, differences in the lamina propria. Okay, if you see the lamina propria, simple tubular glands are there, the intestinal glands, along with that numerous lymphatic follicles are there, which is extending into the submucosa, just like the ileum. Okay, just like the ileum. Okay, so here we don't call it as Peyer's patches. Okay, here we don't call it as Peyer's patches. Here lymphatic follicles are there. Okay, I know there is a confusion why we are not calling it as Peyer's patches. There is a confusion that we will discuss in lymphatic system chapter. Histology of lymphatic system. Okay, so numerous lymphatic follicles are there and that extend into the submucosa. Okay, smooth muscle layers are there, inner circular, outer longitudinal. Now another important point, that is in case of muscularis externa. In muscularis externa, जो longitudinal layer है, which is forming the tinea coli, that tinea coli is absent in appendix. Okay, tinea coli is absent in case of appendix. In submucosa, lymphatic follicles are also extending into the lamina propria. Now here you can see the diagram. In this diagram you can see this is the lining epithelium. Ye dekho, this is the lining epithelium. Then these are the intestinal glands. Okay, intestinal glands. This is the lamina propria where you can see the lymphatic follicles. Okay, diffuse lymphatic tissue, lymphatic follicles, all those things can be seen. Okay, and the quantity is huge. Next, this is the submucosa. Ye dekho, this is the submucosa. Now submucosa glands are not there as usual, and then this is the muscularis externa, and then serosa. Appendix is a intraperitoneal organ which is covered by a fold of peritoneum, that is called as mesoappendix. Okay, so that's why completely it will be covered by serosa. So you can see the serosal layer. So that is all about the large intestine.